Alright, let's get back to our question of how frequently does this actually happen? So, what are my chances of getting just any cancer? You can have cancer that starts in all different cell types. And so a really important thing an important disclaimer is that we can't entirely predict. And that's because, well, we're just not there yet with the science, but even still, there's always environmental concerns, right? And so, even if your DNA says that you should grow up to be a tall and, blonde-haired person, maybe you, what you eat and the environment that you live in prohibits you from doing that. Maybe you don't get enough nutrients, so you don't grow to be very tall, or something like that. So there are definitely environmental contributors. But with the genetic component, we can leverage that to make some predictions. Whoops. Right? So normally a cell replication halts until the DNA can be repaired. So this goes back to the idea that DNA makes mistakes when it's being replicated. You have this DNA polymerase and it makes mistakes sometimes. But there's also this thing called mismatch repair, and it's supposed to come back and repair any of those mistakes. And that's what those two checkpoints were in the cell cycle that we talked about. We've got these checkpoints, right? And in these locations, it says, okay, hold on, let's fix all the mistakes. And so the DNA error rate is estimated to be about 1 in 10 to the 7th. So that means, right, this is 1 over 2, 3, 4, 5, 6, 7. So 10 million, 1 in 10 million times. So that's a pretty large number, right? You have to copy 10 million base pairs before that DNA polymerase is going to make a mistake. And that's just the regular error, right? But then if we have with mismatch repair, right, this is when it comes back and says, okay, hold up, and it's going to fix the mistakes, right? Now it's actually 1 in 10 to the 9th, right? And so that's 1 in a billion. And we only have, we have 3 billion base pairs in our genome. So that means on average it's going to make maybe 3 mistakes every time it copies, and that's just based on these numbers. And so let's do that math real quick. So we have 3 billion base pairs in the genome. So 3 billion base pairs per genome, right? And we're going to multiply that by the error rate, and that's 1 error per 1 billion base pairs copied, right? And that's if we're using mismatch repair. So 3 billion base pairs per genome times 1 error per 1 billion base pairs, and so these can cancel. And what we end up with is 3 errors per copy. And that's just an estimate based on the numbers that we used here. Right, so about 3 errors every time you copy the entire genome. But how many times do we copy it? Well, this happens every time that a cell replicates or a cell divides, and that varies widely. So, for example, for an embryo, that can replicate in 8 minutes. It can complete the cell cycle in 8 minutes. But an adult liver cell, that can take over a year before it actually replicates, and some cells are even longer. So there's a wide array of how long it takes for a cell to divide and how often they do. And clearly the more that they replicate, the higher chance there is that they're going to have errors, at least errors that are a result of DNA polymerase not performing correctly. Now there are a couple of things to note here. First, 3 errors, that's probably not going to make a big difference, but these errors accumulate. So over your lifetime, errors accumulate. And that's what can really cause them to be harmful. And that's what can lead to cancer. And this kind of goes back to that multi-hit hypothesis that we were talking about. Out of these 3 errors, you're not all of a sudden going to have 6 mutations in the genome and that result in those 6 hits and lead to cancer. That's not going to happen just in one copy of the genome. But when this happens many, many, many times over your lifetime, that's when these errors can accumulate and cause mutations. And ultimately those mutations can accumulate and lead to cancer. That said, so the second thing is that not all hits or errors, not all base pair hits are deleterious. And deleterious just means bad. And as we talked about before, only roughly 2% of the genome is coding, right? And that other 98%, it's doing stuff, but it's not actually building proteins. So these hits, there's a really good chance that they're not even going to end up in the coding region of the genome. And if they don't end up in the coding region, then they're not going to be able to cause deformed proteins, which lead to that, the altered signaling pathway, and ultimately maybe one of those phenotypes that's necessary for the multi-hit 6-hit hypothesis. So that's another thing to keep in mind. And even if it does hit one of those genes, that 2% of those base pairs overall, it still might not even have an effect, as we discussed in the last video. So the body is really, it's an amazing thing, and over time it's evolved to minimize your chances of developing cancer.